Welcome to Freddy's BioEd. Today I'm going to talk about a recent pandemic that has put the world on a lockdown. Everyone in the world is talking about coronavirus, right from the most powerful people in the world to presidents, prime ministers, heads of state, local panchayat officers, administrative officers, or to even small children. Illiterate people are also aware of corona, and pretty much everyone is afraid about corona. As human civilization arose, several diseases struck them down. In the realm of infectious diseases, pandemic is a worst case scenario. Open any news channel, it's mostly on corona. It's everywhere. Although everyone is talking about corona or using the word corona, there's a lot of information that you might want to know. There are a lot of unanswered questions. Please stay tuned till the end. You might not want to miss any of the information that I'm going to pass on. I have divided my presentation into 4 sections. Number 1, I'm going to talk about the history of the past. Number 2, biology of corona. Number 3, clinical assessment based on symptoms, the diagnosis and treatment of corona. Number 4, some interesting facts about corona. Let's look at some of the various pandemics humankind has encountered in the past. Let's start with the first one, which happened during the year 430 BC. The earliest recorded pandemic, which happened during the Peloponnesian War. It passed through Libya, Ethiopia, Egypt and Athens. It was caused by Salmonella, which is commonly called as a typhoid fever. Next, we have the Antonine Plague, which happened during the year 165 to 180 AD. Measles was caused by measles morbillovirus and smallpox caused by variola major. Then we have the Plague of Cyprian, named after the first known victim, the Christian bishop of Carthage, which happened during the year 251 to 266 AD. This was also caused by measles or smallpox. Number 4, the Plague of Justinian, which happened during the year 540 to 750 AD, which was called the bubonic plague caused by Yersinia pestis. Number 5 and 6, the Black Death and the Third Plague pandemic, which happened during the year 1331 to 1353 and 1885, was also caused by Yersinia pestis, called the bubonic plague. Now let's look at some of the fundamentals. 3 terminologies that you might want to know before we get started. Epidemic refers to a sudden increase in the number of cases of disease above what is normally expected. Number 2, outbreak carries the same definition as an epidemic but is often used to describe a more limited geographic event. Next one is pandemic, refers to an epidemic that relates to a geographical spread, and it is used to describe a disease that affects a whole country or the entire world. I have one more definition about reproduction number. It's a very important threshold that is related to transmittability of the virus, which is usually expressed as R0. R0 can be implicitly defined as the average number of secondary infections produced by an infectious person. It is generally believed that if R0 is greater than 1, the number of infected cases will increase exponentially and cause an epidemic or even a pandemic. The R0 of SARS-CoV was found to be between 1.4 and 6.9 with a mean of 3.28, which is really high. Many pandemics have occurred throughout the history of mankind. Primarily, plague, smallpox, cholera and Spanish flu are the longest lasting, repetitive and caused large number of human deaths. In the beginning of the 20th century, 20 million people deaths were estimated due to the 1918 Spanish flu. The first originated place of the pandemic is not clear, but many experts assume that the possible origin was in China. Spanish flu pandemic is reported to spread in 3 waves. The first wave in spring of 1918, which was moderate but spread rapidly. The second wave was in autumn 1918, was completely severe and destructive. The third wave was in the spring of 1919, which was more severe than the first wave but was not worse than the second wave. Many countries experienced the second and the third waves of more virulent forms of infection. It is estimated that 50% of the world's population were infected. However, 25% of the population only manifested clinical signs and symptoms. After almost 40 years of the Spanish flu, a new influenza strain was detected in China, which is called as the Asian flu. The 1957 pandemic was caused by H2N2 strain. The virus spread to Hong Kong, Singapore, Taiwan and Japan. and in summer spread to the world in the summer of 1957. The pandemic affected approximately 40 to 50% of the people, and 20 to 30 of them had clinical signs. Most of the deaths were due to secondary bacterial pneumonia. The 1957 Asian influenza pandemic estimated the basic reproduction number R0 was 1.8, and 60 to 65% of the infected individuals were manifested with clinical symptoms. After a decade of occurrence of the Asian pandemic flu, the new flu strain H3N2 caused a new pandemic known as the Hong Kong flu in 1968. Although the new virus was extremely transmittable, the degree of severity was milder than the Asian flu. Mortality of Hong Kong flu was 16.9%. It spread through Vietnam War veterans returning to the United States. Then Hong Kong flu was seen in Japan, England, Wales, Australia, and Canada in 1969. At this pandemic, the characteristic mortality shifted towards the younger population. The case fatality rate was highest among children. Hong Kong flu is estimated to have caused 2 million deaths worldwide in 2 waves, with about 34,000 deaths in the US alone. Then we had the Russian flu in the year 1977 to 1978, which was caused by H1N1. In the April of 2009, a new pandemic occurred, which was caused by H1N1, called the swine flu, was a causative agent which emerged from Mexico, and it was estimated that 8,520 to 17,620 deaths in the US alone. The global trade and travel served swine flu to spread to as 122 countries in 6 weeks. H1N1 pandemic profile had a course with 3 waves in spring, summer, and the fall. Pattern was generally mild wave in the spring and early summer, but re-emerged with more severe intensity after the opening of the schools. Studies showed that adults older than 50 years of age were less susceptible to the 2009 H1N1 infection than younger adults. While compared to the previous pandemic, the infectivity was higher among children than among adults. After the 3 influenza pandemics of the 20th century, humankind did not encounter a new influenza pandemic but a novel coronavirus pandemic. The recent severe acute respiratory syndrome coronavirus SARS-CoV, called the COVID-19, is responsible for the pandemic, causing fatal atypical pneumonia. As we all know, coronavirus disease 2019, COVID-19, originated in the city of Wuhan, Hubei province, China. The COVID-19 pandemic currently sweeping around the world reminds us all of our vulnerability to the enemy that we cannot see. Let's look at some of the biological information about coronavirus. Coronavirus belongs to the order Nidovirales of the family Coronaviridae and belongs to the subfamily Orthocoronavirinae. There are about 4 genera within the subfamily Orthocoronavirinae, namely the alpha coronavirus, beta coronavirus, gamma, and the delta coronavirus. 2019 COVID is a novel lineage of the beta coronavirus, which is closely related to the bat SARS-related coronavirus. The recent outbreak of viral pneumonia was caused by the beta coronavirus. It also caused the severe acute respiratory syndrome SARS and the Middle East respiratory syndrome, the MERS. In 2002, an outbreak of the SARS was first reported in China and then spread quickly worldwide, resulting in hundreds of deaths with 11% mortality. In 2012, MERS first emerged in Saudi Arabia and subsequently spread to other countries with a 37% mortality rate. In both these epidemics, the virus likely originated from bats and then infected humans through other intermediate animal hosts. The civet cat for SARS coronavirus and the camel for MERS coronavirus. Since December 2019, a number of patients with pneumonia of unknown etiology emerged in Wuhan city, Hubei province, central China. They shared a history of exposure to the Hunan seafood market in Wuhan, and the disease was been confirmed to be an acute respiratory infection caused by a novel coronavirus. Genome sequencing has demonstrated that this coronavirus genome is an enveloped positive sense single-stranded RNA molecule. The SARS-CoV-2 genome is 26 to 33 kilobase pairs in size, the largest genome of known RNA viruses. It encodes a large non-structural polyprotein which is called as the ORF1a/B. The ORF1a/B is proteolytically cleaved to generate 15 to 16 proteins, out of which 4 are structural proteins and 5 are accessory proteins. The 4 structural proteins consist of the spike surface glycoprotein, the membrane protein, the envelope protein, and the nucleocapsid protein, which are essential for SARS-CoV assembly and infection. The SARS-CoV-2, upon entry into alveolar epithelial cells, replicates rapidly and triggers a strong immune response, resulting in cytokine storm syndrome and pulmonary tissue damage. It spreads from person to person through respiratory droplets, through coughing or sneezing. It can also spread through contact, including hand-to-hand communication and followed by self-inoculation. Infectious respiratory aerosols at a short distance is also one means of transmission from person to person. Let's look at some of the symptoms a person can have. COVID-19 patients have some gastrointestinal symptoms including fatigue, fever, dry cough, myalgia, and dyspnea. They can also have nasal congestion, headache, running nose, sore throat, vomiting, and diarrhea. The incubation period is about 3 to 7 days for a short term and 2 to 14 days for a long transmission period of the SARS-CoV-2. Now, the detection of SARS-CoV-2. Rapid acute detection of SARS-CoV-2 is essential to control the outbreak of COVID-19. SARS-CoV-2 genome is available at the gene bank, thus the nucleic acid of SARS-CoV-2 can be detected by RT-qPCR or by viral gene sequencing of nasopharyngeal and oropharyngeal swabs, stool specimens, sputum specimens, and blood samples as well. In February 3, 2020, the CDC developed a testing kit for COVID-19, which was followed by the FDA, which authorized the first home-based test kit for detection of SARS-CoV-2. Then in India, in the month of May, 18 different testing kits were made available for sale in India, but still under consideration. I have added the links onto which you can log on and check for yourselves. Now let's look at the therapy that is given to patients affected with coronavirus. The general treatment is antiviral therapy, which has 4 or 5 different types. Lopinavir, which is a protease inhibitor. Ribavirin, a nucleoside analog, which has a broad antiviral activity. Chloroquine is a widely used anti-malarial and autoimmune disease drug. Recently, chloroquine has been reported as a potent broad-spectrum antiviral drug. Still, there's a lot of speculation towards this drug. Arbidol, antiviral drug against influenza infection that was widely used in Russia and China. Remdesivir, the nucleoside analog, was reported to inhibit SARS-CoV and MERS-CoV in vivo. The next type of therapy is cellular therapy. Natural killer cells or NK cells are important immune cells necessary for defense against microbe-infected, stressed or malignant cells. Whilst developing a new drug, new vaccines or clinical trials of old drugs carrying out NK cell therapy to enhance immunity is currently a very feasible strategy for the treatment and prevention of COVID-19. Immunotherapy. Convalescent plasma therapy has been widely used in infectious diseases such as poliomyelitis, influenza A, and Ebola. This convalescent plasma could theoretically be a promising option for the treatment and prevention of SARS-CoV-2 infection, although clinical trials are still going on to check the feasibility of this treatment. Finally, we have the monoclonal antibody treatment. Remission of the SARS-CoV-2 epidemic may depend on the development of monoclonal antibodies. Previous studies have identified a number of effective monoclonal antibodies that target the SARS-CoV spike protein to prevent the virus from entering host cells. Now I'd like to take you to some of the latest updates on corona status, which we can get from few important websites. Number 1, COVID-19 Worldometer. This is a very interesting website which was developed by 2 students at Carnegie Mellon University. This website provides a simple, interactive way to visualize the impact of COVID-19 around the world. The data from Worldometer's real-time updates utilizes reliable sources from around the world. The website pulls new data every 2 minutes and then gives you a real update of the current status. There is another website called as the COVID self-checker page of the CDC. This works for individuals only in the US. For those who live in India, there is an app called as Arogya Setu. The app communicates with nearby phones using Bluetooth and gathers the GPS location data. By using this app, it is possible to keep track of all the people who come near or are in contact with a person who has been infected with coronavirus. The government has launched the Arogya Setu app in early April as a voluntary application, with its use being encouraged by the Prime Minister of India. Latest scientific and public health information is available at Center for Disease Control and Prevention (CDC). It is also available at the website, which is maintained by the World Health Organization. The point I'm trying to drive here is that everyone is fighting to save human lives. Let's not be indifferent to the efforts taken by every country. Every president or prime minister is doing their best to save their country. Me and the Indians salute the efforts of our Prime Minister Modi ji for taking the present condition very seriously and has saved thousands of lives. May God grant him wisdom to tackle the situation. Whichever part of the globe we belong to, you are important. You mean something to someone. We cannot lose this fight. Let's win this together. Let's save the human species. I got a few more interesting information. First known use of corona. The word corona was in the year 1563. I could not find out why this word was used, but I got this information from the Webster's Merriam Dictionary. Another interesting information is there was a wagon called as Corona wagon by Toyota in the year 1971. Another piece of information, Corona Typewriter 3, which was also available probably during the same period of time. There is also a Corona City Hall in California. There is a Corona Arch known as a Little Rainbow Bridge in the US. There is also a football player by name Jesus Manuel Corona. The best one is there is also a beverage called as Corona beer. Thank you for listening to this presentation.